Place a human subject in a functional MRI machine and instruct them to chant Om. Within minutes, the scan reveals a rapid, localized shift in blood flow. The activity in the amygdala dims completely. Deep within the temporal lobe, the amygdala acts as the brain's primary alarm system. It constantly scans the environment for threats, governing the onset of the physiological stress response. But during the vocalization of Om, metabolic activity in this specific node drops sharply. The threat detection center effectively powers down. This imaging confirms an objective, measurable suppression of neurological reactivity. The change occurs within the brain's physical hardware, separate from the practitioner's internal reporting. The brain's default resting state, characterized by constant low-level vigilance, is actively overridden by a deliberate physical behavior. To trace this override, we must look past the spatial maps of an fMRI and examine the electrical data at the millisecond scale. This chart tracks event-related potentials, or ERPs. It measures precise electrical spikes generated when the brain processes visual stimuli. Normally, when looking at a neutral image, the waveform spikes sharply up and down, but during Om chanting, this entire waveform flattens. The P1 spike, representing early visual attention, drops. Milliseconds later, the late positive potential, tracking cognitive appraisal, also shows a severe reduction in amplitude. The visual cortex receives the exact same visual data, but the baseline reactivity to the outside world is suppressed. This electrophysiological dampening is the result of a highly specific biological sequence. To understand it, we need to map the mechanical and neurochemical pathways engineering this exact state. The practice operates on two strict mechanical parameters. First, it requires an acoustic fundamental frequency typically ranging between 100 and 150 Hz. Second, it demands an extended, controlled exhalation. Steady, observational. This precise combination of sound and breath has been maintained by monastic practitioners for thousands of years. Prolonging the expiratory phase while vocalizing creates continuous physical friction. This low-frequency vibration mechanically stimulates the receptors embedded throughout the laryngeal muscles and lung tissue. Those receptors trigger an immediate afferent signal. An electrical impulse fires upward along the vagus nerve, climbing out of the chest and toward the brainstem. The signal arrives at the medulla oblongata, the anatomical region responsible for autonomic control. This targeted stimulation of the medulla forces the body out of sympathetic arousal, the biological fight-or-flight mode, and locks it into parasympathetic dominance. The physiological metrics shift instantly. Blood pressure drops, and heart rate variability steadily increases. The chanting protocol uses controlled, localized physical vibration to force a systemic, bottom-up deceleration of the human nervous system. Once the vagal signal reaches the brain, it initiates a specific neurochemical cascade, starting deep within the thalamus. This diagram illustrates the cellular synthesis of gamma-aminobutyric acid, or GABA. This is the central nervous system's primary inhibitory neurotransmitter. The incoming vagal stimulation upregulates a specific cellular mechanism. Inside the presynaptic terminal, glutamic acid decarboxylase accelerates the conversion of glutamine, first into glutamate and finally into GABA. These molecules are packed into vesicles and released via exocytosis directly into the synaptic cleft, heavily increasing GABA concentration in the thalamic relay stations. The thalamus is the brain's primary routing center for sensory data. Flooded with GABA, it restricts the volume of external information allowed to pass through to the higher cortical regions. The brain physically tightens its sensory gates. This localized inhibition synchronizes thalamic oscillations into the 8 to 12 Hz alpha range. This rhythm restricts early visual attention, which is the exact biological mechanism responsible for that flattened P1 electrical spike on the ERP scan.
The practice acts as an endogenous anxiolytic. It chemically blocks external noise from reaching conscious awareness. Simultaneously, the vagus nerve sends projections into another critical brainstem structure, the nucleus tractus solitarius. These projections directly stimulate the raphe nuclei. This small cluster of neurons acts as the main production hub for serotonin, triggering a widespread release of the monoamine throughout the central nervous system. This visual maps out a serotonergic synapse. For serotonin to effectively alter a person's conscious state, it has to bind to highly specific receptor subtypes scattered across different brain regions. Neuroimaging shows that chanting heavily increases binding at the 5-HT1A receptors, actively suppressing the release of cortisol, mitigating the body's chemical stress response. At the exact same time, there is a marked change in activity at the 5-HT2A receptors. The 5-HT2A receptors are deeply integrated with the brain's default mode network. Modulating this network alters introspective awareness, quieting the brain's running internal monologue. Serotonin acts as a system-wide dial. It stabilizes the baseline emotional state while physically rewriting how the brain observes its own thoughts. The brain only manufactures about 10% of the body's total serotonin. The other 90% is synthesized entirely within the gut, managed by the enteric nervous system. Look at the physical mechanics of the chanting process. It requires deep, prolonged abdominal breathing, forcing the diaphragm to repeatedly expand and compress. This rhythmic pulmonary action applies direct physical pressure to the gastrointestinal tract below. That persistent physical massage of the gut tissue stimulates the localized enteric networks, driving a massive spike in peripheral serotonin production, which communicates back up to the brain via the gut-brain axis. The practice recruits the entire peripheral nervous system, enforcing systemic calm through these direct biological feedback loops. If the brain's sensory gates are chemically restricted and its threat detection node is deactivated, a structural question emerges. Does this state compromise necessary evolutionary survival instincts? When researchers introduced a highly aversive, threatening image to practitioners actively chanting Om, the flattened ERP waveform violently spiked. Despite the dense GABAergic blockade and the serotonergic buffering, high arousal negative stimuli instantly shatter the artificial calm, capturing full attentional resources. The amygdala reacts immediately to genuine danger, stripping away the neurochemical inhibitors to prioritize immediate survival over relaxation. The practice functions as a selective biological filter. It mutes the exhaust of baseline resting anxiety without blinding the organism to actual real-world threats. These immediate neurochemical shifts occur within minutes of chanting, but maintaining the protocol over weeks produces a second, chronic effect. Consistent activation of the GABA and serotonin pathways triggers the release of brain-derived neurotrophic factor, or BDNF. This protein acts as the primary catalyst for the growth of new neurons and synapses. This map illustrates the specific neuroanatomical processing zones targeted by this neurotrophin signaling. Longitudinal MRI scans track this growth. Over a 12-week period, novice practitioners develop a measurable increase in gray matter density across the hippocampus, the prefrontal cortex, and the insula. This structural reinforcement provides a tangible buffer against chronic stress. Adding physical tissue to these regions reinforces the brain's baseline resilience. At the molecular level, DNA methylation patterns begin to shift. Within weeks, the genes responsible for triggering inflammatory stress responses are downregulated. The vocalization of Om is a precise, repeatable mechanical protocol that structurally reinforces the architecture of the human brain.